Hello, my name is Leslie Kenny. I'm one of the co-founders of the Oxford Longevity Project, and today I'm here with two of my fellow co-founders, Dr. Paul Chen, and with Professor Dennis Noble of Oxford University, and we are very privileged to have in our presence Professor Yoshimori from Osaka University and Dr. Ishido, also of the Japan Autophagy Consortium. And Professor Yoshimori he's instrumental when it comes to mammalian autophagy research. And Dr. Ishido is helping with the Japan Autophagy Consortium, which is an academic, government, and industry group to really solve this problem of aging. So we have a lot of questions for them, and we hope you enjoy this discussion. Dennis?
So, my interest in interviewing you is that following the discussion with Professor Osumi, we looked at the big achievements he had made in relation to autophagy in yeast. And my understanding there is that autophagy in yeast is essentially a single cell process for getting all the detritus that has come into the vacuole out of the system.
Yes.
But it doesn't kill a cell, at least not normally. Whereas in the mammalian immune system, there's a big difference. That is that it is actually looking to find cells that are no longer functioning properly and to ask them to apoptose. Now, you've been involved in the mammalian system, and what would be interesting to know is how similar are the two? To what extent can we extrapolate from what was discovered in yeast to what needs to be done in the mammalian system?
I see. So essentially, autophagy system itself is very, very conserved one, and so most of ATG genes Osumi Sensei found are also in mammalian. So that's I think a basement of autophagy study, and of course there are many differences, but essentially system is similar to yeast. Mammalian autophagy.
So specifically, the genes involved seem to be similar, don't they?
Yeah.
So how is it that during evolution it's transitioned from autophagy in a single cell, with the vacuole being the process, to autophagy applied to cells in a big population that need to be brought out? Do we have any understanding of that, or is that a big issue still?
Yeah, of course, during evolution, autophagy itself is evolved. For example, in yeast, autophagy doesn't have a role for cell protection, kind of immunity.
Yeah.
But in mammals, autophagy can be a protection mechanism against invading pathogen. So this function does not exist in yeast.
Indeed.
So,
that's right.
During evolution, mammalian use autophagy for different function. So that's a big difference. Not only immunity, but also others, many kind of function. Have been added there.
Yes, indeed.
During evolution. I think.
One of those must be the ability of the system to make its genome change in relation to the coding for immunoglobulins when the system is invaded, because clearly in contrast to what happens in yeast. But there's my question: is there something comparable when the immune system in the case of an invasion by a new virus, a new bacterium, will literally start to change the sequence in the variable part of the encoding for the immunoglobulin. And presumably that's a new development. Am I right? That does not exist in yeast?
Yeah, I agree. Yeah.
No.
But basically, I think system is quite similar. So from yeast to human, so because probably autophagy is so important for life. So then system is conserved, but we can apply the system to other function. Yeah.
But clearly the question of exactly how all of that evolved is open still, isn't it?
Yeah.
That's an extraordinarily
still there is,
difficult question.
Yeah, many.
Yes. Well, I wonder whether, shall I hand over to you now? And do you have the points we were going to raise, or shall I pass this sheet over?
Why don't you pass them to me?
That's exactly.
Good. Right. So, for instance, we talk about yeast and how they're similar to mammalian cells, but some cells in the body don't actually replicate so frequently. I'm talking about cardiac cells. And so how important is autophagy to maintenance of the heart and the cardiovascular system?
Yeah, so if you knock out autophagic gene in cardio muscle cells, then you have a failure of cardiac system after aging. So investigator in Osaka University published already then. So, not only cardiac cell, but also central nervous system. In nervous system, neuron doesn't change during the whole life in human. So then, also it's so important. Even cell change, for example, skin cell change, but still autophagy is important, but more and more important in long life cells like neuron and the cardiac system. So in cardiac system, you have heart failure if you don't have autophagic activity there. In neuron, you have Alzheimer-like syndrome. Of course, it's an animal experiment, but we know all mouse has so Alzheimer-like phenotype if you knock out autophagic essential gene, ATG gene in nervous system.
So I have a question as a patient. What can patients do, or what can the general public, what can we do to activate autophagy safely in our bodies?
Safely, yes. Of course, we should develop a drug for autophagy activation, but I think so ordinary custom life, what you say, life custom, it's okay in English.
Habits, practices, lifestyle habits.
Yeah, lifestyle, yes. For example, aerobic exercise. Aerobic exercise, okay. Induce autophagy, and good sleep induce autophagy, and less calorie induce autophagy, and several food which contain some factor inducing autophagy actually exists, so you can take a good food. For example, natto.
Spermidine.
I know. Some European doesn't like natto, but natto has a lot of spermidine So if you choose a good food, I think you can induce autophagy. Especially, we found that autophagy decline with age. Aged people, I think, need more this kind of custom. But if you start this habit or lifestyle earlier, I think it's better.
Of course, it's better.
Because you can't do this suddenly. So longer time, style change. From a younger period is so important, I think.
And if natto can induce autophagy, or if spermidine can induce autophagy, does Western processed food increase Rubicon?
We don't know yet. You mean, we know that some lifespan extension intervention, for example, calorie cut, and for example, mitochondrial little bit suppress like mitochondrial function. So we already know several lifespan extension invasion. All of these inventions showed Rubicon decrease. So this means that this kind of treatment or lifestyle can decrease Rubicon, I think.
By extension, my question to you, Professor Yoshimori, the Rubicon obviously is a negative regulator of autophagy, correct me if I'm wrong. And of course you have the other types of products like spermidine or rapamycin. Do you think there will be other types of negative regulators? And why? Obviously, it seems to be a very fine balance between both sides, and of course, whether it's upstream or downstream. Do you think that we will find a situation where we can, for lack of a better word, manipulate it so that we get the right balance to enable us to live longer and healthier?
Yes. I think so. Not only Rubicon, but also other factor control autophagy. And if you enhance autophagy other than Rubicon inhibition, still I think it does work, even if you have a lot of Rubicon. But for example, several reagent or several food contents induce autophagy independent to Rubicon inhibition. So balance is important. Even if Rubicon suppress autophagy, you can induce autophagy by other way.
And what is your views? I mean, from my own background, I'm quite interested in cancer. And I know that autophagy is slightly tricky. It's like a double-edged sword, as things are. And of course, in terms of the early phase of cancer development, it seems that autophagy has a protective effect. Whereas in the latter stage, it seems to potentially have a sort of a detrimental effect. Of course, there's a lot of emerging science coming out of that. Again, similarly, do you think that we'll be in a situation clinically down the line where we can then map out what sort of processes are going on within the cell to then in the tumour milieu or in terms of physiologically, we then be able to manipulate with various feed products or, for instance, from the pharmaceutical industry to have sort of certain chemicals that we know will guide through various metabolic pathways or ones in which we then specifically direct autophagy within the cells in terms of the immune cells that we need, your NKT cells, your T cells, your B cells, versus actually you want a negative regulator, for instance, in the situation with the cancer cells.
So I think only cancer is difficult to be treated by autophagy. I mean, so for preventing cancer growth, you need inhibition of autophagy in cancer. But as you said, immunity for cancer For example, recently, Professor Honjo, Nobel laureate in Kyoto University, found that the spermidine enhance cancer immunity. So in aged people, so a bit difficult. But I can say, so some people ask me that we shouldn't have food inducing autophagy if you take care cancer, but I don't think so, because even you have small cancer inside cell, and you didn't notice this, but I think enhancement of autophagy doesn't affect cancer growth. Inhibition of autophagy is even so. Autophagy help cancer growth, but already enhanced the cancer, I think. It's my personal opinion. We don't have evidence, but I think so. So cancer required autophagy, but in these cancer cells, already cancer is autophagy is enhanced, so autophagy inducer doesn't affect more cancer growth, I think. You understand my
absolutely.
It's difficult to explain, but inhibition is required, but enhance doesn't affect, so not to enhance cancer growth, I think. So then, but so tumorigenesis, autophagy prevent tumorigenesis. We already know. So generally, I think autophagy enhancing is beneficial. But if you have growing cancer, so at this stage, probably inhibition is better. But in future, we think, I think, targeting, we can do this. So autophagic inhibitor only go to cancer, and for other tissue, for example, so immune system, autophagy enhancer, so separate way is better. So
I guess it must be very difficult, because clearly with autophagy you have apoptosis, and then you have the antigen presentation side of things, and that interplay is very much a fine balance. So it would be quite interesting to see down the line that interplay.
Yes.
I've got a question for Dr. Ishido. You've been working with academia, business, and government. And have you found a lot of support within government to promote autophagy to the Japanese public at large?
I think so, yes, they support us a lot. We got a grant from NEDO, 経済産業省っていうの what's the it's a kind of government society, and they granted us to support make some the guideline and rule about the supplement foods, which has the contains the ingredient that has the autophagic enhancement activity. So it's autophagy is not so popular still in Japan, but it's getting popular, and already we can see some foods that says autophagic activity, but actually we're not sure about that. So we like to make it clear which supplements is actually conducting proper research to see they have autophagic activity or not. So our consortium is trying to set such kind of guideline, and the government supports about that project. Yes.
And what about medical doctors? Do you work together with hospitals and medical societies to teach them how to work with patients to activate autophagy?
I think that's a really important activity, and that's exactly our next step. And and Yoshimori sensei is discussing exactly what we should do, collaborating with clinical physicians and to do some service to measure the autophagic activity and make big data about that.
Wow, that's very exciting.
Yeah.
Now, I know that some traditional Kampo medicines contain ingredients with molecules that have been shown to activate autophagy. So curcumin and nobiletin in shikawasa both also activate autophagy, and they're part of Kampo. And yes, so I wonder to what extent you would work with a group like a Kampo medical society to create protocols around autophagy activation. In Japan, I know that Kampo medicine and Western medicine, they work side by side. It's not like in the United States or the UK.
Actually, my colleague in Osaka University, Professor Takeshi Noda, he is studying Kampo, and he published one paper about one of Kampo contents are enhancing autophagy. I think by collaboration with one Japanese Kampo company. They have a library of but the problem is Kampo, so Chinese medicine, very complicated. And it's quite difficult to identify the molecule level. I mean, so you separate Kampo, then function disappear. So probably not one molecule, but
mixture.
Is important.
Yes, I think that must be the case, actually. I have a bit of experience of excuse me a moment. I have a bit of experience of working on Kampo myself, and it is possible to do some good physiological analysis of how a Kampo medication is working. But I think you're right to suspect that trying to find single molecules that will do the same thing is going against what Kampo is based on. Because after all, it's based on plants. Now, plants have had around 300 million years to work out how to convince us to eat them, because that's how they distribute their seeds, how they get to be spreading from one part that they can't walk after all. So I think the millions of years of interaction with other organisms that can move has led to them refining the combinations that seem to work. So I think we just benefit from that. There's no point in splitting it up, because they've already found out it's necessary to have it together.
Yes.
Now, I mean, it's difficult though, because possibility of clinical trials, difficult. I was involved myself in trying to convince a major company here in Japan to go to clinical trial, given what we'd found for the product that we were working on. But once they found the cost, they retreated and didn't go any further.
Well, I think this has been a really productive talk and a great day. Thank you again for your help in organizing the interview, the discussion between Professor Osumi and Professor Noble, and also arranging the Autophagy Consortium this evening. We look forward to many years of continued collaboration. Thank you.
Yeah. Thank you.